from the last video we are learning about the PCR. We have learned about the basic principle of the PCR, okay, and also we have covered the primer designing. Today's video, it is concerned about the inverse PCR, which is a type or the variation of the PCR. This inverse PCR, it is also called as the inside out PCR. Inverse PCR, it is used for the amplification of the DNA whose sequences they are unknown. So here you have an unknown DNA sequence. Suppose this it is a genome, and on this genome this it is a particular unknown DNA sequence, and this unknown DNA sequence when you want to amplify, at that time you are using the PCR, and the type of the PCR that you are going to use, it is the inverse PCR. But when you are planning to do the inverse PCR, at that time with this unknown DNA sequence you should have some of the sequences which are known to you. Okay, the flanking DNA. Okay, the flanking DNA, it should be known to you. Okay, then you can have the amplification of the DNA. Now when we have learned the principle of the PCR, at that time what we have learned, this it is a double stranded DNA, it is a template DNA. Then this template DNA in the denaturation step, it is getting denatured, it is forming the single stranded DNA. Next step, it is the annealing, and in the annealing the primers they are, okay, the primers they are going to anneal to the template DNA, and in the extension there is a polymerization and the daughter DNA strands it is going to be formed. And this step from the extension, again there is a step of the denaturation, annealing, then extension. This cycle it will go on and on for about the 30 times, and then you are getting the amplified DNA product. Now here when you are saying that you are adding a primer, okay, the reaction it is in vitro. Okay, so this primer you have to design, you have to synthesize this primer, and for the synthesis of this primer there is a need to have the information about this template DNA. Okay, for the designing of the primer, what is required? Information about the template DNA, that is the DNA sequence of the template, it should be known so that you can design a primer. Without the template DNA information you cannot design a primer. So whenever you say that you have to amplify the DNA, you are using the primer and you are amplifying the DNA, at that time you should know something about the DNA sequence. So here when you are saying that you have to amplify, okay, you have to amplify these unknown DNA sequences, at that time near about there should be a known DNA sequence so that you can design this primer. Okay, so that you can design this primer. So for the designing of the primer, here there is a need of the known DNA sequence which is near, which is adjacent to the unknown DNA sequence. Okay, so here there is this unknown DNA, with that there is a known DNA sequence. But this DNA, where it is present, this DNA it is present over the genome. Okay, so first you have to amplify. Okay, no, not amplify. First you have to isolate. Okay, first you have to isolate this part. Okay, this part of the DNA, a small piece of the DNA you have to first isolate. For that purpose, you are going to do the restriction digestion. Okay, for that purpose what is required? Restriction digestion, and in the restriction digestion you are using the restriction endonucleases. Type 2 restriction endonucleases, or the restriction enzymes of the type 2 you are going to use. Here, when you are going to select the restriction endonucleases, when you are selecting the restriction endonucleases, at that time what should be kept in the mind? That restriction site, okay, the restriction site, it should not be in between this DNA. unknown as well as the known DNA sequences. That is, here it is an unknown DNA sequence and this it is a known DNA sequence. So your restriction site, it should fall where? Outside this region. That is, it should flank the unknown and the known sequences, so that when you are going to do the restriction dilatation, this part it will be intact one. Okay, second thing, when you are selecting restriction endonucleases, it should produce the sticky ends. Sticky ends here. These overhangs, okay, these overhangs, they are called as the sticky ends. These overhangs, they can complementary base pair and it can form a circular DNA molecule. Okay, so here the restriction endonucleases 2, when you are using its restriction site, it should flank the DNA that you want to amplify, and the second one, it should form the sticky ends. Okay, so after restriction dilatation, there will be the number of restriction fragments. Okay, and from that restriction fragment, this particular restriction fragment you have to isolate. For that purpose, you have to go for the isolation and the purification of the DNA, and with the help of the southern blot, you are going to confirm, okay, or you are going to identify this particular sequence, and then that DNA molecule, it is going to be go for the amplification by the inverse PCR. Okay, so you have isolated the restriction fragment, which is consisting of the unknown DNA sequence with the known DNA sequence. Now here you have to, okay, here you have to design a primer, and that primer, it is going to be designed, okay, the primer, it is going to be designed for whom? For this known DNA sequence in this way. Okay, and this it is a restriction fragment. Okay, so here the primer, it is going to be designed for the known DNA sequence. After restriction dilation, there is a generation of what? There is a generation of the sticky ends. Then you have to go for the ligation. Okay, so after ligation, you are getting the double stranded DNA. Okay, what you are getting? You are getting the double stranded DNA. Okay, understanding? Okay, so this, it is a unknown DNA. Okay, and the remaining, it is the known one. Okay, so here, now imagine this, it is going to circularize. So one primer, okay, one primer, it will be in this direction. Okay, and the second primer, it is going to be in this direction. Okay, so there is the use of the two primers. Okay, and two primers, they are going to face in the opposite direction. Okay, that is here it is a 3' OH, here it is a 3' OH. Okay, and if you can see, they are in the opposite direction, and when it is a conventional PCR, at that time this 3' OH and this 3' OH, it is how? It is in the same direction. Means the here, okay, here it is inverse, it is happening here. Here it is going to amplify the DNA which is in between the two primers, and here it is going to amplify the DNA outside, okay, at the outside. So it is an inside out. Okay, so there is an amplification of the DNA, and after amplification of the DNA, you are getting the two DNA strands, and that two DNA strands, they are going to be a linear one. How they are? They are the linear DNA strands, and then like the PCR there will be the amplification of the DNA. In this way there is a procedure for the inverse PCR. In this way there is the PCR, inverse PCR. Now by amplifying the unknown DNA sequence, what you are going to achieve? That is, what are the applications of this inverse PCR? First application of the inverse PCR, it is in the chromosome walking. Okay, where it is in chromosome walking for the preparation of probes. Okay, so for the preparation of the probes in the chromosome walking, inverse PCR it is going to be used. Next, it is used to identify the insertional site of the viruses, transposons and the transgene, and it is also used to amplify the unknown cDNA from the mRNA. So these are the applications of the inverse PCR. Now here suppose here it is the identification of the insertional site for the viruses, transgene or the transposon. Suppose this it is an insertional sequence, and it may be a transposon, or it may be a viral DNA, or it may be a transgene, because you know the sequence of the transposon, you know the sequence of the viruses, you know the sequence of the transgene. So that it is a known sequence, and what you have to find out where it has been inserted. When this unknown region you have to find out, and this it is a known region. So what you are going to do, you are going to prepare the primer for, suppose if this is a transposon, the sequences from the transposon you are using for the primer synthesis, or it is a transgene sequence, it is also known to you. If then you are going to design a primer. If it is a viral DNA sequences, they are known to you, then with the help of that known DNA sequence you can design a primer, and in this way you can amplify the insertional sequences, okay, for the viruses, transposons and the transgenes. And after amplification, okay, after amplification of the DNA sequences, you can do this DNA sequencing, and you can find out which are the bases which are present at that particular site, and in this way you can identify the insertional sequences, insertional site for the viruses, transposons and the transgenes. I think you may have cleared the inverse PCR, its principle, its application. If you have any doubts, if you have any comments, you can write down into the comment box. And also, if you like my video, please subscribe my channel and share my videos with your friends, so the knowledge it is going to spread. Thank you.